Hi students, last class we talked about mitosis, and now I want to give you a brief overview of meiosis. Mitosis and meiosis are two stages of the cell cycle. So for most cells in our body, the cells will go through interphase and then go into mitosis. And mitosis produces identical cells. And so mitosis is used for growth and tissue repair. Mitosis can also be used for asexual reproduction in single-celled organisms. So a bacteria, for example, a bacterium will go through its cell cycle and then at the time of reproduction, go through the stages of mitosis and make duplicate copies of itself. For us, our cells go through mitosis whenever we grow, and if we get a cut on our hand and we need to repair that tissue. Meiosis, on the other hand, only happens in the testes of males and the ovaries of females. That's for humans. It's different organs with plants to make gametes. And so gametes come together in sexual reproduction. So meiosis is used just so sexual reproduction can happen, so that sperm cells can be produced and egg cells can be produced. First, we're going to look at mitosis and meiosis compared. Then we'll look at an overview and the stages of meiosis, and then we'll talk about the human life cycle and how it relates to mitosis and meiosis. So for mitosis, we have the division of the nucleus one time. So the cell goes through interphase and the nucleus goes through that division process of mitosis. That's followed by cytokinesis and the splitting of the cytoplasm. This produces two daughter nuclei. Each daughter nucleus has the same genetic material. Remember in mitosis that the sister chromatids separate, and that's identical copies of DNA separating. So what was present here is present here. This preserves the chromosome number and produces genetically identical daughter cells. They're identical to each other and to the original cell. Meiosis, on the other hand, involves two divisions of the nucleus. So the nucleus divides one time and another time. This produces four daughter cells and cuts the chromosome number in half. We'll see how that happens in a minute. The daughter cells are genetically different from each other. So each of those four cells is different, and each of those four cells is different from the original cell. Cuts the chromosome number in half. And this is used only for producing gametes for sexual reproduction. So an overview of meiosis. This is a special type of cell division just to make gametes, cuts the chromosome number in half. And if we think about what's happening with the human life cycle, it kind of fits in and makes sense. And we're going to look at this pictorially at the end. So a parent is diploid. So the cells in our body, except our sperm cells and egg cells, are all diploid. That means chromosomes are in pairs. Remember those homologous pairs? That's what I'm talking about. Diploid can be abbreviated as 2N. Meiosis produces haploid sperm cells and egg cells. Those types of cells are called gametes. And the abbreviation for haploid is N. So it cuts the chromosome number in half. When gametes fuse in fertilization, the N plus N makes 2N. Simple math. Brings the 23 chromosomes from a sperm cell to the 23 chromosomes in an egg cell. 23 plus 23 is 46. This produces a diploid, now 46 chromosomes, zygote. That's the first stage in an organism's life. This produces the next diploid generation. So that fertilized egg, now diploid, will undergo mitosis for growth. This picture shows very simply what happens in meiosis. The parent cell at the top of this diagram has 4 chromosomes in the nucleus. So the nucleus is the blue area. There are 2 long chromosomes and 2 short chromosomes. The long chromosomes, even though they're not next to each other, represent a homologous pair. They're similar. And the short chromosomes are the second homologous pair. So this parent cell is diploid, has a 2n number or diploid number of 4 chromosomes. The DNA duplicates. That happens in the S phase of interphase. Okay? And interphase happens the same for cells that will undergo mitosis and cells that will undergo meiosis. So we have the G1, the S phase, and G2 happening the same way for these cells. The DNA has replicated and now it looks like little X's in the nucleus. These are the sister chromatids held together by the centromere, and the sister chromatids are identical to each other. So what's sticking together in that X? They're identical chromosomes. In the first nuclear division, the homologous chromosomes separate, but the sister chromatids stay together. So on the cell on the left, you see a long X and a short X. And on the cell on the right, a long X and a short X. So the longs separated and the shorts separated. Those are the homologous chromosomes. In mitosis, the cell division separates the sister chromatids as the centromere dissolves. That happens in the second nuclear division. And so at the bottom, you can see that the cell with the long X and the short X splits, and we end up with a cell with a long chromosome and a short chromosome, and another cell with a long chromosome and a short chromosome. And the same thing happens to the cell on the other side. This produces 4 daughter cells. They're haploid. They have half the number of chromosomes as the parent cell, and they just have one of each type, a long and a short. So they're genetically different from the parent cell, clearly, but they're also going to be different from each other. As I said, the cell cycle always starts with interphase G1, S, and G2. Okay, in G1, the cell grows, recovers from the division, synthesizes some of the proteins it needs. In the S phase, the DNA duplicates, and in the G2 phase, the cell prepares for division. Then mitosis or meiosis occurs, followed by cytokinesis. Now we'll look at the steps of meiosis. And you'll see meiosis 1 at the top of this slide. That means that we're looking at the first division in meiosis. So all these steps happen as we separate the chromosomes for the first nuclear division. Meiosis 2 involves the steps for the second nuclear division. So this starts with prophase 1. And you'll be happy, I hope, to hear that the steps of meiosis have the same names as the steps of mitosis. But you'll see there's a number after each step. So prophase 1 is the first step in meiosis 1. Okay? It needs to be that specific because what happens in each of the steps is slightly different. In prophase 1, duplicated homologous chromosomes pair up. So they're condensed, the nuclear membrane starts to dissolve, the nucleoplasm starts to disintegrate. We'll even see the spindle start to form. But what's different here is that the duplicated homologous chromosomes are right next to each other. They even overlap. So the homologous chromosomes here are the short X's and the long X's. The 2 short X's are the homologous pair, and the 2 long ones are the other homologous pair. They've duplicated, so the X's mean they're sister chromatids. In metaphase 1, homologous pairs align independently at the metaphase plate. So this is fairly similar to metaphase in mitosis, where chromosomes line up at the plate, but here they are lined up side by side. You can even see that something has happened in prophase 1. There's been an event called crossing over, where the ends of the chromosomes overlap, they cut off and reattach themselves. So now all the chromosomes, the sister chromatids, are different. Next comes anaphase 1, where homologous pairs separate and move to opposite poles of the cell. Note that the centromeres haven't dissolved. We still have sister chromatids intact, but the homologous pairs have separated. In telophase 1, the daughter cells have one duplicated chromosome from each homologous pair. This cell will split in two at this point. After telophase 1, there is no interphase. The cell goes right into meiosis 2, or may have a resting period called interkinesis. What's important here is that there's no replication of the DNA. In prophase 2, after interkinesis, that resting period, the cells have one duplicated chromosome from each homologous pair. These are the separated cells. Then we go into metaphase 2, where the chromosomes align at the plate. And I know my picture is in the way there, but both cells look identical. This is exactly what the cells look like in metaphase of mitosis. In anaphase 2, the sister chromatids separate, exactly like mitosis. That involves the dissolving of the centromere and the pulling apart of the sister chromatids by the kinetochores attached to the microtubules. In telophase 2, the spindle disappears, the nuclei form, and we end up with 4 haploid daughter cells. In the life cycle, mitosis and meiosis alternate with each other. And this happens with more time in plants and humans. Let's look at humans first. You'll see in this picture we have 2 adult individuals almost at 9 o'clock here, male and a female. Meiosis happens in the ovaries of the female and the testes of the male, and that causes us to have haploid gamete cells, the sperm cells and the egg cells. They're haploid, that means 1 N, or just simply N. When they come together in fertilization, that means we have 2 N together. That's diploid. And that diploid individual, the zygote, which develops into an embryo, to a young individual, to an adult, grows through the process of mitosis. I would say, if I could relabel this picture, from fertilization to meiosis is mitosis. In plants, it's a little bit different. You can see the plant there where the adults are, adult humans on the other picture. In plants, we can have meiosis happening in 2 different parts of the flower, producing sperm cells and egg cells. Those will be haploid. And the male cells, the male gametes, sperm, will fertilize the female eggs through various mechanisms. If you take a botany course, you can learn about the different types of plants. Some plants need sperm from other plants to fertilize the eggs. Some plants can self-fertilize, and some flowers can even self-fertilize. Once the fertilization takes place, the plant goes through mitosis just like a developing human zygote. I hope that helps with your study of meiosis. Be sure to differentiate between mitosis and meiosis and know the details of both.